Hi, in this video I want to talk briefly about G protein coupled receptors. So I want to go just basically over the structure and then sort of how they're going to work. And so let's begin. So here we've got a G protein coupled receptor. So the receptor is right here. It's got these 7 transmember alpha helices. So I just represented them with lines. And so you're going to see that in basically all G protein coupled receptors. Now, this is not a channel, so when you get its ligand, so say this is acetylcholine, and it binds to this, it's not actually going to go through. It's going to stay on the outside, but it's going to have an effect on something that this receptor does bind to, which is the G protein. That's why we call it a G protein coupled receptor. So acetylcholine binds, and then it's going to do something to this G protein, specifically this alpha portion, the yellow or I guess orange one that I have here. And this is a Gtp binding protein, which is where the G comes from. And so what it's going to do is it's actually going to release a GDP that it has and take up a GTP, and it's going to split that is going to cause a conformational change that will split the alpha section from these beta and gamma sections. So the beta and gamma sections will go over here, the alpha section will go over here, say to the right. And each of those is going to initiate an effector system that will then have an effect on the cell. So for example, it could initiate an effector system that ends up releasing calcium into the cytoplasm from a vesicle, as an example. So there will be different types of effector systems, and I'll make a video about those. But for now, just understand that a G protein coupled receptor induces a change in this alpha, beta, and gamma, and these alpha, beta, and gamma subunits of the G protein, which then go and cause 2 different effector systems or cascades to operate.